Bismillahirrahmanirrahim. Welcome, my dear students, senior one, to a new biology session. Today, inshallah, we'll continue discussion on the unit 1 in biology, chapter 2, chemical structure of living organisms. Today, inshallah, we're talking about proteins and nucleic acids. In the last session, we revised together, we have studied together carbohydrates and lipids as examples of biological macromolecule. Okay, and today, inshallah, we are gonna study proteins and nucleic acids. Okay, just give me a minute. Now we are going to study proteins and inshallah nucleic acids as examples of biological macromolecules in our body. Our body is made mainly of 4 types of biological macromolecules: organic molecules, carbohydrates, lipids, proteins, and nucleic acids. Proteins. Now we have to talk about proteins. They represent the structural composition of all living organisms. Most of our body is made of protein. If you see your body, many parts in our body and many organs are made of protein. Okay, and proteins make huge portion of or ratio of the body of living organisms. They are biological macromolecules made of smaller molecules or monomers called amino acids. Okay, proteins are very important in all biological processes in our life, in our body. All biological important processes in our body need proteins. You have to know, for example, most of hormones are made of proteins, and most of enzymes are made of protein, and muscles in our body made of protein. The blood contain a protein, a certain protein, and the liquids, many liquids in our body may have proteins, and some parts of the body like nails and hair, and in animals there is the hooves or the horns in the animals are made of protein also. So protein made a big percentage in our body and in the body of living organisms. Protein are very important. They are the basic components of the cell membrane. They are main components of the cell membrane. And proteins form muscles. Proteins form muscles. Also, protein help in formation of muscles. The basic components of the cellular membrane, and they make the muscles and the ligaments, the tendons. الليجمنتس اللي هي الأربطة والتندونز الأوتار in your joints. The gland nails, the glands, الغدد, the nails, and the hair. A lot of vital fluids of the body made of protein also, like blood and lymph, made of protein. So they are necessary for bone growth. Furthermore, enzymes and hormones are made of protein, which regulate the vital processes in the body are also protein. So proteins are the basic component of the chromosomes also. So proteins are the biggest percentage of biological macromolecules in our body is a protein. For example, the network made by the spider. The network made by spider is made of protein. The horns of some animals made of protein. The hooves made of protein, and so on. Proteins very important. Here we have how the proteins are formed. The protein molecule is a big molecule or a polymer. Polymer of protein, we can call it another name, polypeptide. Polypeptide. It is made of a long chain of smaller units or monomers. These monomers are called amino acids. So many amino acids join together to make what? To make the molecule of protein. Many amino acids join together by polymerization process, and they make a peptide bond. So the molecule resulted is called polypeptide. This is the protein, the polypeptide. Okay. So we have the protein is made from combination of many units, smaller units called amino acids. But is the question here, what is the amino acid? What is the monomer or the building unit of protein? We have here an example of amino acid and the general formula of amino acid. This is the general formula of amino acid. We have carbon atom in the middle. We have a basic element which is carbon in the middle of the molecule. Carbon is the middle or the center atom of the molecule. And we have 4 bonds. We have 4 bonds. One bond with carboxyl group. One bond with carboxyl group, COOH. Carboxyl group is acidic group. Acidic group. And another bond with amino group, which is NH2, and it is basic. So we have acidic group and the basic group. So the amino acid or the protein are very sensitive to pH, the pH value. And we have also another bond with hydrogen atom and another bond with alkyl group. We mentioned it R. This is for R. These are for alkyl group, a group of carbon and hydrogen, many carbon and hydrogen atoms. Like you can say C2H2, C3H3, and so on. Alkyl group. This is the structure of amino acid. The basic structure of amino acid. The structure of amino acid, protein building units, they are organic compounds made up of hydrogen, oxygen, and carbon, in addition to nitrogen. We have here also nitrogen, which is found here in the amino group. Nitrogen also is one basic element in the amino acid. We have in the figure here the shape of the amino acid, the basic group, amino group NH2, and the acidic group, carboxyl group, COOH. These two groups are the functional groups, the functional groups of the amino acid or the protein. Hydrogen atoms is a terminal, and also alkyl group is different from one amino acid to another. So the difference between amino acid and another amino acid, not in amino group, not in carboxyl group, not in hydrogen, and not in carbon, but the difference between amino acid and another amino acid here in the alkyl group. So how many amino acids in our body? How many amino acids in our body? The scientists calculate the number of amino acids in the body of living organisms. They found them only 20 amino acids. Only 20 amino acids. But they join together to make a long chain of polypeptide. They join together to make a long chain of a polypeptide. So the polypeptide chain is made of amino acid. How many amino acids? Not the 20 make one protein, but maybe 10, maybe 2, maybe 3, maybe 5, maybe 6. So there is difference in the number of amino acids. And also there is difference in the arrangement, the order of the amino acids. By changing the order of amino acids, we can make a new protein. By changing the number of amino acids, we can make a new protein. So how many protein can be produced? We can produce infinite number of proteins, very large number of proteins. Although we have 20 amino acids, but we can make a big number of a very big number of proteins by changing the arrangement or the order of amino acids, and by changing the number of the amino acid and changing their order and changing their type. We can change the type of amino acids, we can change the order of amino acids, and we can change the number of amino acids. So we can produce large number of proteins from only 20 amino acid. We can produce big number of protein. So we have only 20 amino acids, but we have a large number of proteins. How can amino acid join with another amino acid? There is a bond formed between two amino acids. This bond is called peptide bond. It's joined. The joining happens between the carboxyl group and the amino group from another amino acid. The carboxyl group of amino acid, like this one, and the amino group of another amino acid, like this one. What will happen here to make a chain of two amino acids? We say it's called dipeptide molecule. How we can make a dipeptide molecule? By removing water, removing two hydrogen atoms and one oxygen atom. By removing what? By removing two hydrogen atoms and one oxygen atom. How? We remove one hydrogen from amino group, from amino acid, and we remove hydroxide group OH from another amino acid. So they make one water molecule, H2O. We can remove one water molecule, H2O, by removing two hydrogen atoms and one oxygen atom. So by removing one water molecule from the two amino acids, we can make the bond, the peptide bond between the two amino acids, and we make a new molecule made of two amino acids. We can call it a name bipeptide, or dipeptide molecule, dipeptide molecule. We can make three also by making another removing of another water molecule. We can join another one here by removing carboxyl hydroxide group from one carboxyl group and removing H from another amino group. We can make another bond here between a third amino acid. So by removing water molecule, we make the peptide bond. By removing a water molecule. So we have here how the peptide bond is formed. You can observe this bond are present between carboxyl group. This one is carboxyl group COOH, and the amino group NH2 of amino acid, and the amino group NH2 of another amino acid. By removal of water due to this combination. So the formation of bond need removing one water molecule. It's called dehydrolysis process. The product of a combination of two amino acids is called dipeptide compound, and the protein chain formed of several amino acids is called polypeptide chain. Many amino acids join together, they make a polypeptide chain. سلسلة عديد البيبتيد. But only two amino acids join together, they make dipeptide molecule. مركب ثنائي البيبتايد. It's not a conditional. The combination occur among similar amino acids. No, it's not a condition to bend between two similar amino acids. They can be different. This gives extensively wide and various possibilities to form a protein. Depending on what? Depending on the type, and the order, and the number of amino acids. the order, the type, and the number of amino acids, we can make a very big number of different proteins. We have only about 20 amino acids participate in building the protein chain, such as glycine, alanine, and valine. These examples of simple proteins. These examples of simple proteins: alanine, valine, glycine. They are made of binding of some amino acids. Now we are going to classify amino acids into two groups. Classify proteins into two groups: simple proteins and conjugated proteins. Simple proteins and conjugated proteins. What is the difference between simple proteins and conjugated proteins? The simple proteins are made of basic units of building units. Building protein, amino acids only. Amino acids only. Such as what? Example of simple protein, we have albumin protein, which is present in the leaves and roots of some plants and in the plasma of the blood of the human. Present in the leaves and roots of plants and in the blood plasma of the humans. This is an example of a simple protein called albumin. We have another type of protein called conjugated proteins. Conjugated means it's connected or joined with another element. In addition to amino acids, there is additional elements added, such as phosphorus or iodine or iron. When these elements are added to the chain of amino acids, they make a conjugated protein. The meaning of conjugated: bonded with another element. We have the nucleic protein, the conjugation associated with what? With phospho and phosphoprotein. Conjugated protein also, like the protein associated with the nucleic acids. They are called nucleic proteins. These called nucleic proteins. The meaning of nucleic proteins: there is a nucleic acid joined with amino acid. Nucleic acid with amino acids. These make nucleic protein. And phosphorus joined with amino acid, such as casein protein, which found in the milk. Casein protein found in the milk. Conjugated amino acids are conjugated with what? With phosphorus elements. So it's called phosphoproteins. It's called phosphoproteins. And we have also the milk protein, such as casein. We have phosphorus element. And we have also thyroid protein or thyroxine. If you remember, we studied in grade 9. Thyroxine is a hormone secreted from the thyroid gland. هرمون الثيروكسين secreted from thyroid gland. يفرز من الغدة الدرقية. This hormone, the amino acids are joined with what? Joined with iodine element. Joined with iodine element. And the last example, hemoglobin is a protein also joined with iron element. Hemoglobin is found in the blood, in the red blood cells. We have a protein called hemoglobin. Hemoglobin is a conjugated protein because it is joined with iron element. So we have conjugated proteins such as what? Such as nucleic protein, such as phosphoproteins, such as thyroid protein, such as hemoglobin. All of these are conjugated proteins. The amino acids are joined with other elements. Nucleic acid joined with protein, it's called nucleic proteins. Phosphorus is added, it makes phosphoproteins. Iodine is added, it makes thyroid protein, thyroxine. And iron is added, it makes hemoglobin found in the blood. This is the conjugated proteins. Now we can move to the nucleic acids. We remember that we have two types of nucleic acids. DNA and RNA. You remember we have studied DNA in grade 9, which carry the hereditary material, carry the genes. It's found in the chromosomes, and they carry the hereditary substance or the genes. You remember the chromosome, the shape of the chromosome like this. The chromosome has a shape like this, like letter X. And there is here a point of connection called centromere. And we have here inside each nucleotide, we have 2 chromatids. Each chromatid has a molecule of DNA, a long molecule of DNA, a long molecule of DNA. This is the structure of the chromosome. This is the structure of the chromosome. If you remember, we have studied it in grade 9. This is the structure of chromosome. It is a long thread of DNA molecule, the nucleic acid DNA, which is symbolized for deoxyribonucleic acid. Deoxyribonucleic acid. And we have also RNA, which is symbolized for ribonucleic acid. And we'll study the difference between DNA and RNA. The difference here, this is deoxy and this is ribo. Deoxyribonucleic acid and ribonucleic acid. Ribose sugar here is complete number of oxygen. Deoxyribonucleic acid, there is a lack of 1 oxygen atom in the ribose sugar, so it is called deoxyribonucleic acid. The nucleotide is a basic building unit of nucleic acid. The nucleic acid is also made of units, smaller units. If you imagine the shape of the DNA molecule like this, the DNA molecule like this, there is many units here, the DNA made of many units. These units or divisions are called nucleotides. The building unit of DNA is called nucleotide. So each division or each step here is called nucleotide. What is the structure of nucleotide? The nucleotides are the basic units forming the DNA, the nucleic acid, and also the RNA. It's composed of 3 units, and we'll illustrate them here in this shape. We have a pentose sugar, like ribose. We have ribose sugar in RNA, and we have deoxyribose sugar in DNA. It is a pentose sugar. What is the meaning of pentose sugar? This means it has 5 carbon atoms. Pentose means it has 5 carbon atoms. Join it with what? Join it with phosphate group. Carbon atom number 5. Join it with phosphate group. And also join it with nitrogenous base. Carbon atom number 1, join it with nitrogenous base. Carbon atom number 5, join it with what? Phosphate group, which is PO4. So we have 3 basic structures. 3 basic structures. Pentose sugar, سكر خماسي. In RNA is called ribose, but in DNA is called deoxyribose. Right. This is part number 1. Part number 1, which is a pentose sugar. Part number 2 is called phosphate group. Join it to carbon atom number 5. Part number 3 is called a nitrogenous base, which is joined to carbon atom number 1. This is a structure of nucleotide. So we have The first type, which is deoxyribose sugar, involved the composition of DNA. DNA is deoxyribose sugar, but RNA is a ribose sugar. We have ribose sugar. So we have one oxygen atom is shortage of one oxygen atom in deoxyribose sugar in DNA. So also we have phosphate group. The structure, we have phosphate group. Number 2 structure, number 2 phosphate group. Join it to number 5 carbon atom number 5. And the nitrogenous base, we have 4 different nitrogenous bases. In DNA, we have 4 different nitrogenous bases, which are adenine, guanine, G, cytosine, C, and thymine, T. We have 4 different nitrogenous bases. But we have to remember that adenine always comes with thymine, with double bond. Adenine always bend with thymine, with double bond. And cytosine always bend with guanine, with triple bond. We have also A with T, and we have also C with G. It can be TA also. It can be TA. And it can be GC, no problem. It can be GC. But always A with T and always C with G. Come with G. Okay? This is in DNA molecule. In DNA molecule. But in RNA molecule, T is not found. T is replaced. In RNA, T is replaced. In RNA, we have RNA. We don't have thymine at all, but we have another nitrogenous base which is called uracil. So we have adenine, we have uracil instead of thymine. We have cytosine, and we have also guanine. So we have 4 nitrogenous bases in RNA. There is A and there is C found both, and G found in both, but U only found in A, uracil found only in RNA, and thymine found only in DNA. This is one difference between DNA and RNA. The nitrogenous base here may be A or T or C or G, but in RNA the nitrogenous bases may be A or U or C or G. Okay? Nitrogenous bases: adenine A, guanine G, cytosine C, thymine T. This is in DNA molecule. This is in DNA molecule. But in RNA molecule, we have uracil. In RNA, instead of thymine, there is no thymine, and there is a uracil instead of it. RNA have uracil instead of thymine in DNA. Each base is connected to the carbon atom number 1. Connected to carbon atom number 1 here in the nucleotide of the sugar molecule. So sugar molecule has 5 carbon atoms. This is number 1, this is number 2, 3, 4, 5. This is number 5. Nucleic acids differ with respect to the different type of pentose sugar and nitrogenous bases forming them. Different in pentose sugar and also nitrogenous bases. Pentose sugar in DNA, deoxyribose sugar. Pentose sugar in RNA, ribose sugar. Nitrogenous bases in DNA, ATCG. Nitrogenous bases in RNA, AUCG. We have here the shape of DNA and the shape of RNA. The difference in shape between DNA and RNA. You can see that DNA is double thread. double thread, not one single thread like RNA. RNA is a single thread. RNA is a single thread, but DNA is double thread, double helix. It's called double helix. Double thread. The DNA is double thread, but RNA is single. Single A thread. This is the first difference. The first difference between DNA and RNA. DNA always found inside the nucleus. Doesn't go outside the nucleus of the cell, but RNA is produced inside the nucleus and can move outside the nucleus of the cell to the cytoplasm. The RNA is a part of DNA, a copy of a part of DNA. So DNA is very long thread, RNA only a part of DNA, a copy of a part. Like we take this part and make a copy, we make RNA and we send it to the cytoplasm. We send it to the cytoplasm. So the DNA make a small copy of part of it, which is the RNA. The DNA contains the genes which are responsible for appearance of the hereditary traits. They are found on the chromosomes inside the cell nucleus. They are responsible for passage of the genetic traits from one generation to another when the cells divide. You remember the mitotic division and the meiotic division. The chromosomes are divided and go to the new cells, carrying the DNA, carrying the hereditary traits, carry the genetic information responsible for appearance of distinctive characteristics of the living organism and organize all vital activities of the cell. The genes or the hereditary characters found in DNA and also RNA is a transcribed from DNA. يعني a copy, transcribed, يعني a copy of DNA. Then it transfers into the cytoplasm. It is made in the nucleus and transferred to the cytoplasm. But DNA doesn't move to the cytoplasm. To synthesize what? The protein responsible for appearance of genetic traits. So the order is carried with RNA. The order found in DNA, but it is carried by RNA to the cytoplasm to be formed. Responsible for organizing the vital activities of the living organism. So DNA and RNA are the most important molecules, biological macromolecules in our body. We come to the end of this part of the in chapter 2. We come to the end of the part of chapter 2. We have here examination how to detect the proteins. Detect the proteins. We have here an reagent called Biuret reagent. Biuret reagent. We can bring some samples. We can bring some samples of different food material such as egg, albumin, starch solution, sucrose solution, distilled water. Which of them contain protein? You think? Egg or starch or sucrose or distilled water. Which of them contain protein? Starch. Which egg or starch or glucose or sucrose contain protein, which help in building the body? Of course, it is what? It is
egg albumin.
Of course, it is egg albumin. Of course, it is egg albumin which contain the protein. The meat, the eggs, the milk, all of these contain protein. And we have a reagent called Biuret reagent. Biuret reagent. If we add the reagent to these samples, which of them gave a positive result? Only one of them, the egg albumin gave a positive result. Only the color changes into violet in only the egg albumin. The color of Biuret changes into violet. or purple. This is a positive result. A positive result that means that you can test the presence of protein by using the Biuret reagent. Which is negative? Starch is negative, saccharose will be negative, distilled water will be a negative. The color of the Biuret reagent doesn't change. Compare between observation and prediction. You can say the change in the color means that the egg albumin contain protein. This is a test for the proteins. Come to the end here. We have some questions I want you to solve if you can, and send me. I will see them inshallah in the next session. In the next session, inshallah, we'll go to chapter 3. We'll go to chemical reactions in organisms' bodies. After chapter 3, we can make a complete revision on unit 1. Inshallah, we'll finish the study of unit 1 completely. Then we make a session for revision on unit 1, inshallah. Okay? Have any question about what we have studied today?
No.
Thank you. I
No.
hope you understand. I hope you studied very well. Inshallah. Until we meet again, thank you and goodbye.